So I can introduce Alan. He'll go first. So Alan Milby was a student at Virginia Tech. He graduated in 2019, I think, with a degree in wildlife conservation. But he has a strong appreciation for plants too, and he worked in the Massey Herbarium with us for the last year or two of that time. He was also in our Natural History Collections Club as one of the founding officers there. And he had an interest in the smaller components of the forest. So for his project, he studied the mosses and lichens growing in stadium woods and also in center woods nearby. So Alan, he graduated a couple years ago, and since then he's been working at the Harvard University Herbarium as one of the technicians there. So he's still pretty involved in plants, and tonight he'll give an update on his stadium woods work. So Alan, you can feel free to take it from here and share your slides.
All right. Well, thank you for that, Jordan. Very nice. You make me sound pretty cool. All right. Let's see if I can share my screen here. Beautiful. And of course, this is blocking this. All right. Well, hello, everyone. Very glad to be here. So as Jordan said, this is a project that he and I started when I was an undergraduate at Virginia Tech. And the title is Comparing the Cryptogam Floras of Old Growth and Secondary Growth Forest Stands on the Virginia Tech campus. So for those of you who don't know the word cryptogam, as probably most people do not, that comes from the words or the roots crypto, meaning hidden, and gam, meaning gamete. So pretty much talking about spores. So organisms that reproduce by spores. So this here is a picture of an old growth forest. And I really like this picture. I really get a nice serene feeling from it. This haze sifting through the trees, these big white oaks, and that open understory, very characteristic of old growth forests. But little would you know that this was actually taken on the campus of Virginia Tech, actually in a forest right behind the football stadium. And it's called Stadium Woods, very well named. So as Stadium Woods is like most other old growth forests that are left in the United States, what is left is what is considered urban old growth forests. They're fragmented with urbanization all around it, or they're kind of restricted to these mountaintops and mountain ridges where mechanical equipment couldn't get in to harvest it. And this is very sad because they're really great habitats for a specific suite of species and organisms that require the stability that old growth forests provide for them. So particularly for cryptogams, like I mentioned, in our case, lichens and bryophytes, they are dispersal limited. So their spores don't move around very easily. So in an old growth setting where that's very stable across time and across space, they can really move about the forest very easily and diversify, because since old growth forests, have that antiquity, they have a lot of microhabitats and niche spaces for these things to diversify in. Yeah. So I thought I'd give you guys, seeing as how this is a native plant society, I thought I'd give you guys a pretty good introduction to lichens and bryophytes. Very quick and dirty, because there's a lot you could talk about for both of these organisms, many of lectures you could do. But hopefully by the end, maybe you guys can start noticing these things out in the field and know a little bit more about them. So I'll start here with lichens. So they're considered a symbiotic complex that has a fungal partner and a photosynthetic partner. So there's a fungi and then a partner that does photosynthesis. So that's either algae, normally green, that's the most common partner. Sometimes there's brown algae, that's much less common. And then cyanobacterium is another partner that can be part of the lichen complex. So there's a lot of contention about the type of relationship that the fungi and the photosynthetic partner have. But I think it's been most accurately put as a type of controlled parasitism where, the fungi is getting, these, photosynthetic compounds, like sugars and carbohydrates from that algae. And it kind of buries into the algae cells to do that. But the algae a lot of times cannot survive in the environments that they're in without that fungal component. So it's a very interesting relationship that they have. And I thought I'd go ahead and try to show you guys the different forms, the major forms of lichens. They're really in a huge variety of forms. It's what really grasped me at first. So we have on the left, crustose form. So these forms are normally on rocks or bark, and they're fused with their substrates. So you can't take this lichen off of where it's growing. It's intermixed between the particles. So normally, if you're out with a lichenologist and they're in the field and they're collecting crustose lichens, you can hear them off in the distance, they'll carry a hammer and a little pick, and they'll be chipping off lichens from boulders. That's always pretty fun. So then we have the foliose forms here, which, is kind of frilly, kind of foliose like a leaf. And they are not fully fused with their substrate. Normally, you can take these off of what they're growing on pretty easily. And then on the far right, we have a fruticose form, which is kind of this 3D form. And the picture shows what is commonly called, I think, British soldiers. I think some of you might be pretty familiar with those. So that form is considered fruticose. So the diagram on the right-hand side here kind of shows what's going on within the lichen a little bit better. And it's showing you a cross-section of what's called the thallus of the lichen. So you have the upper cortex, the algal layer that has the algae and, the hyphae of the fungi invading it, then the middle layer, which is the medulla, and then that lower cortex. And then the bottom is what's holding the lichen to its substrate. And this is a foliose lichen, a cross-section of one. So lichens are considered early successional species. So they are the first to get to areas. They also exude certain acids that break down rocks. So they kind of are the starters of soil and our soil base, which is very cool. And they are very sensitive to air quality. So because of that, they've been used as bioindicators. So in their fungal hyphae there, they will trap heavy metals. And you can even do tests to see what heavy metals are in the area by taking lichens and I guess, grinding them up and doing various tests on them. I don't know how to do that. They're also very useful as these bioindicators because they're very sensitive. So only some lichens will not grow in areas where there are high pollutants. You only get a certain number of species that are tolerable of high pollution. So you can do field studies and collect lichens and see how much or what species are there to see how healthy the forest is. They're also used as old growth indicators, as I mentioned, because they are in the beginning, because they're dispersal limited. They've been used to test the antiquity of forests and stuff like that before. So then we got to our next group, the bryophytes. So these are your mosses, probably the one most people know about. But then there are liverworts, which are also called hepatics, and then the hornworts. And these are land plants that lack a vascular system. So like a tree has xylem and phloem to move nutrients and water throughout the plant, bryophytes do not have that. They normally use external features, either hairs or other small features on their leaves or stems to move water across their surfaces. Their leaves are normally only about a cell thick. So they also can kind of absorb that water and nutrients directly through their leaves and stems as well. And on the left here is a really cool liverwort. It's called Palavicinia lyellii. All right. There we go. All right. So on the right-hand side, I wanted to show you guys the two major portions of a moss or a bryophyte, which is the gametophyte, which is the leafy portion, and then the sporophyte, which is that long thing up there that releases the spores. So in regular land plants or angiosperms, the flowering plants, they are sporophyte dominant, whereas bryophytes are they're gametophyte dominant. And the gametophyte is just responsible for producing those the gametes, the sperm and the egg to create the embryo. So they're different from their other land plants because of that. So I mentioned they are an ancient lineage of early land plants. So they're really thought to be kind of the stepping stone that plants took from water onto land, or at least somewhere in that range. And they're very sensitive to microhabitat changes. So as I mentioned earlier, that's why they really like that stability of old growth forests. And that's also why they've been used to indicate forest continuity. Just another way of saying things that have been on the landscape for a long time or are continuous spatially. So contiguous forest. And then I thought I'd show you guys the multiple forms of liverworts. So these are all liverworts. And there's three major growth forms that you'll get in them. So on the left, we have the simple thalloid liverworts. They have that central branch and kind of this single cell layer of tissue along that. Very cool. I think they're very cool. And then in the middle, we have what is called the complex thalloid liverworts. So you guys may have seen these around different streams or wet places before. They kind of look like snake skin. Then they kind of have that appearance because they have these air pores on top of them. And these are obviously multiple cells thick. They kind of have this top layer of chloriferous, has chlorophyll in it, and cells that do photosynthesis, photosynthetic layer. And then the middle has it's packed full of these what are called hyaline cells. So they're basically empty cells that are used for storage, particularly water. Oh, sorry. And then on the right, we have leafy liverworts. So as they sound like, they're leafy. They have individual leaves. And I chose this because sometimes you find really cool things in your liverwort samples, like a tardigrade. And I was very excited to find this tardigrade. You can find leafy liverworts on the bark of trees and all over the place, damp logs, stuff like that. And I didn't put a picture of a hornwort in this presentation because we didn't really have any on our in our study sites. But you guys should look up a picture of hornworts because they are a really cool organism. If you guys don't know what they look like, I wish I had put a picture in. So our study location was in Southwest Virginia in Montgomery County. I'm indicated by that star. I'm in Blacksburg. And this is kind of a zoomed in version of that, showing you Montgomery County in this little outlay here. And Center Woods represented by the circle and Stadium Woods by that star roundabout. And it shows Montgomery County here, and it's in two ecoregions of Virginia, which I thought was interesting. I didn't know that before. So the southern part is in the Blue Ridge Mountains. And the rest is within the Northern Ridge and Valley section. So there's quite a bit of difference in elevation within the county, which is really cool. But our study sites was in the Northern Ridge and Valley section of Montgomery County. So to get a little bit closer, here we're showing you Virginia Tech's campus and both of our forests. So on the right-hand side, you see Stadium Woods, kind of outlined in this burgundy color. A little hard to see, but it's, as you can see, it's really surrounded by all kinds of urbanization, housing complexes. It has the football stadium right next to it, and it has the tennis courts at one end. Very busy. And on the left is Center Woods, showing you our plot that we created within it, which was just of equal size to Stadium Woods to keep sampling the same, keeping the sampling area the same. And it's, you can see it's really just kind of pasture land and agricultural fields right around it. And Route 460 is there on the bottom right-hand side. And there's a few buildings on the edge, but really not much going on. So just some historical context about these forests, kind of just to show that they have been around for a long time. So this, on the left-hand side, we have a map from the Civil War from 1864, and it's showing you center woods on the map in orange and stadium woods by that burgundy polygon. So they've been along at least as long as that. And then on the right is from a quill thesis that was done in center woods, again showing the extent in 1935. So they have been on the landscape for quite a while. So a little bit more about these study sites. So Stadium Woods is about 4.8 hectares, and that equals about 12 acres, roughly. It's dominated by kind of this northern hardwood, and it's an urban old growth forest, as we mentioned. So the species you kind of find in here, the most dominant tree is white oak, and there's these huge white oaks in there that dominate the stand. They date to about over 350 years old. It's really cool to see those trees in that forest. Other species you find in there are black oaks. You'll find sweet cherry and black cherry. You'll also find black haw and a suite of invasive species and stuff like that. So as we saw, it's located right on the main campus of Virginia Tech. And, the picture on the right-hand side is just showing you a game day at Virginia Tech. So all kinds of people, thousands of people, shuffle through Stadium Woods to get to the game each season. So it sees a lot of use by the university. Other than game days, which is a lot of use, Stadium Woods has a pretty old history of use. So this is showing you what was called Hurricane Hill. After World War II, there was the GI Bill, and all the veterans were coming back and going to school. So Virginia Tech put up this trailer camp in Stadium Woods. So those veterans that were coming back to go back to school would have somewhere to stay. And there were some remnants of Hurricane Hill still in Stadium Woods. Other than that, forestry labs and soil labs both go out there. Dendrology labs go out there to do field trips and whatnot. And the Corps Cadets also uses it for training. They have erected a training facility on one edge of the forest. So Stadium Woods is used quite a bit. So then Center Woods is much larger. It's about 17 hectares, which I think is it's 40 something acres. I forget the exact math there, but it's dominated by northern hardwoods, a similar suite of species as Stadium Woods, but it has an oak hickory cover type. So it's has white oaks, and it has some of those large white oaks that we saw in Stadium Woods, but much less frequent. There's only a few speckled throughout the forest. We also get black oaks in here. Northern red oaks make an appearance. You also get shagbark hickories and mockernut hickories, and still some of the other shrubby plants like black haw, sassafras you'll find in here, stuff like that. And so we saw in the map that it was on the outskirts of the main campus, agricultural lands all around it. And here is just showing you a aerial photograph from 2019, because there is some stuff going on in here, but it's mostly used for wildlife field studies. So in the bottom portion of the forest there, in the southern part along the edge, are a bunch of facilities for the wildlife department. So on the left is some storage buildings, and there's an aquaculture center where they do mussel work. There's a research aviary they just erected there not too long ago. There's been some small mammal trapping throughout the stand at different portions of time, and then a quail thesis was done. But otherwise, the stand has seen not that much use, especially not as much as Stadium Woods. and there is Route 460, as I mentioned, on one side. So the big part of this project was that we wanted to compare, because the lichen and bryophytes, could really tell us about the health of these forests. We wanted to compare the lichen and bryophyte flora. So we had predicted that even though stadium woods, it being older and being this old growth stand, that because it was, its disturbance history was much deeper, that it was probably going to have less species and less rich in species than center woods. And now we do have some data, which I'll get to. So first, to get to that question, we needed to collect. So we went out and exhaustively collected stadium woods and center woods, which was, in total, I think there was like 24, 25 acres. Much of what, which I did myself, which was a long process, but very fun. So I went out and collected about 400 samples and recorded habitat data and field notes for each specimen. And that data was then used to create voucher specimens that were accessioned into the Massey herbarium. So this is a picture of me collecting with 2 helpers. And because it was a lot of work, I did get the Natural History Collections Club at Virginia Tech to come out and help me, which was a big help because I would not have finished. I collected between June of 2018 and December of 2018. There's a lot of other stuff going on. But they came out and I trained all the volunteers on basic field identification and collecting for lichens and bryophytes. So they learned, but like what you guys learned today, the different types of lichens, liverworts, how to differentiate between those things, and then how to take good field notes. So we finally got all of our samples and they were in the herbarium. So then we had to do our identifications. So we used primary literature, like this book here, Guide to the Liverworts of North Carolina by Marie Hicks, a fantastic book if anyone wants to get into identifying liverworts. I even use it up here in Massachusetts to identify liverworts, so it's very good. Then we used standard laboratory techniques to identify, standard microscopy. Everything had to be looked at under a microscope. These things need microscopy, so you have to do cross sections of leaves of moss and stuff like that. And most of the species are differentiated by cellular structures. So you really have to look at these things. So pretty time consuming. But we did get some data and everything finally got identified. And now we have some preliminary results. So for our liverworts, we identified 5 species that were unique to stadium woods and 4 that were shared between the forests. And stadium wood had no unique species in it. So in total, that would mean that center woods had 9 species and stadium woods had 4, but they did share 4. And of those, 5 were new to Montgomery County, which was very cool, a very cool outcome of this project to identify some new county records. So on the top right, you see Noelia curvifolia, a very cool little liverwort that's all over decorticated logs in forests all over Virginia. And it has these like really interesting bloated leaves with these minute tips on them. Very cool. And it's showing you a perianth there, the female part of the liverwort. Then on the bottom right, you're seeing Blepharostoma trichophyllum, which was identified in one of our samples, which has these really interesting filamentous leaves. 2 really cool finds. I was very excited about these. So then for our mosses, we had 13 shared between the forests. And then there were 13 unique species found in center woods and then 11 found in stadium woods. So that would give center woods 26 total and stadium woods 24 total species, which was much closer than we would have guessed. And within those, there were 10 new to Montgomery County, which was also very cool to find. And I put just a few of them here. Um, and this map here is the distribution of Physidens bushii, just because I thought it was interesting. Most of the collections have been on the coastal plain and Piedmont regions, but not that much of the plant was documented in west of the Blue Ridge. I think this is the 5th or 6th county west of the Blue Ridge where this plant was found, which is very interesting to find. Really could be just due to lack of investigation, but still an important find. And at the top, you're seeing Physidens taxifolius. I couldn't find a good picture of Physidens bushii, but I thought it'd be cool to show you guys this genus because it's pretty noticeable out in the field. It's called the feather leaf moss, something like that. And it looks, just like a feather. So it's pretty easy to notice when you're out in the field. It's one of the first ones I started noticing, and I really like it. It's a pretty cute moss. All right. So for our lichens, bryophytes were very hard to identify and very time consuming. So we never got to our lichens. So that's still ongoing, and hopefully we're going to get those identified soon. We did create an iNaturalist project to see if we could kind of crowdsource and get some IDs that way. So if anyone knows anything about lichens and would like to, there are photographs of all of our lichens on iNaturalist, and that's the link to get there. Feel free to go on there if you wish. Yeah. So that was about it. So I wanted to give a big thanks to the Metzger lab that I worked with at Virginia Tech because they really helped this project get along, and the Natural History Collections Club for that. And then a group that really funded this project was Friends of Stadium Woods. They gave me a grant so I could do the field work and do some of those identifications. Without that, this project would have never happened. So big thanks to them. Yeah.
Alan, you've got a question in the chat there.
Ask questions. Oh, putty root orchid. Let's see. I do believe putty root orchid was in Centerwoods. I didn't record very many of the herbaceous plants because they didn't have too much to do with the lichens and bryophytes that we were collecting, even though I wanted to do that. And then slime molds, I'm not sure if I did come across slime molds, to be very fair. I'm sure they were out there, but not that I can remember off the top of my head. Oh, and I should also thank Tom Wiebolt because he helped me do a lot of the identifications of mosses. And I'm sure some of you guys know Tom Wiebolt. He did a lot of good work and helped me out with those. Oh, and there's putty root in 2 places in Stadium Woods. Ah, cool. I'm happy to give anyone information on doing any identifications on lichens and bryophytes. I used a lot of different keys for that part of Virginia. So
I have a question.
Yes, go ahead.
Yeah, I wondered, they look as if they'd be hard to put in the collection. So can you tell us how you preserved them for the collection?
Oh, for sure. So they're actually relatively easy compared to plants because you don't have to press them really. Some of the more bulky mosses you do, but really for collecting, I just used a putty knife and scraped them off of their surfaces, whether it was soil or the bark of trees or a decorticated log, rotting log, or whatever. And I stuck them in a little paper bag and then brought them into the herbarium. And they press down pretty easily. and most of them are pretty flat anyways. So you just put them in a packet and they stay pretty safe in the collection in a archival packet. Much easier than you think.
I have a question too. Mary Rose, you remember Chuck Parker? He and his wife used to collect lichens and they put them in matchboxes.
Oh, that's cool.
Yeah. So Stadium Woods used to have lots and lots of English ivy growing up the trees.
Yes.
So would that have inhibited lichens from growing on the trees?
I would say so. I've never thought of that, but I'm sure it would. If it can't get to the bark, then I guess it can't colonize. Yeah, that's a very interesting question.
I've really been there a long time, and they look awfully clean to me. To be,
Yeah, some people did a lot of work out there, taking out invasives, and yeah, a lot of work. Yeah. So, I mean, that's still ongoing. I'm sure it is. But yeah, some people really went out there and did some hard work getting invasives out of there.
Yes, Fred and I just spent 2 hours here today. On Sunday, we spent 6 hours with cadets and all. So, yeah.
So it's you.
Well, us and many other people, but.
Fair enough.
Yeah.
Man, well, that's cool to meet you.
So the liverworts, I always think of liverworts being like in streams or on rocks. When you say there's liverworts, where are they?
So there are ones that will grow on the barks of trees, particularly a genus called Frulania, and you can kind of look at it. It's hard to notice unless you're really looking for them. And that's when I really got excited about liverworts, is when I started being able to notice them on trees. But you can find them on barks of trees in hardwood forests pretty commonly. You also can find them on just downed logs, especially ones that are starting to rot. There's a suite of species that will, genera that will grow on moist logs and stuff like that on the forest floor.
So they're like real small, fibery. I mean, the ones I've seen in, I forget the name of the park, but over there, Montgomery Park. Anyway, I've seen them on the rocks, and they were big, the liverworts. So you're saying they would be really tiny or thread-like?
Yeah, some of them are much smaller than those. You might have seen those complex thalloid liverworts. Those are much bigger. Yeah, you can get ones that are very small. Normally, you need a hand lens to see some of them, or at least see the leaf structure.
We really appreciate anybody that can identify anything in Stadium Woods. It's awesome that you did that.
It was very fun, very hard. And Rebecca Paulson. Oh, Rebecca's here. Yes. Beautiful. She worked with Friends of Stadium Woods. Alrighty. Well, any other questions, guys?
Thanks a lot, Alan. Really appreciate you presenting, and I'm very proud of your work here. So.
Oh, thanks, Jordan.
But if anyone's interested in following up, I can put my email in the chat, and I can put you in touch with Alan, or maybe he wants to throw his in there. But next up, we have some more Stadium Woods research. This is a little more preliminary. This is a project that's just started recently. But this is done by Chrissy Herzog. She's an undergraduate at Virginia Tech. She transferred into VT year and a half, 2 years ago, and is a biochemistry major, but she has a deep interest in fungi, and she's been with us in the herbarium for a year or so now. And Chrissy, I'll let you unmute yourself, and you should be able to share slides and show us what you've been up to.
Thank you for the good introduction. My computer's acting up a little bit tonight, so bear with me. Hello, everybody. So I can get this project up. So my project is the fungi of Stadium Woods. as Jordan said, I am Chrissy Herzog, an undergraduate at Virginia Tech and mushroom enthusiast. I'm working with him on this project. Even though he's not as into mushrooms as me, he's really nice for working with me on it. We're also working in Stadium Woods, and that's the only place that I've collected from so far. So right now, only about 0.5% of the total forest acreage in southeastern USA is old growth, and Stadium Woods makes up about 12 acres of that. It contains mostly white oaks. Some of them are over 300 years old. It's home to many species of animals. It's not a huge topic at Virginia Tech currently, but in 2012, there was some pretty controversial stuff going on with it where people wanted to cut down about 5 acres of it for an athletic facility. And there were protests about this and also about people parking under trees and potentially harming the root systems of these ancient trees. So I think about 5 people were arrested from those protests. And also, if you can see here, it's kind of hard, but this photo shows the Colbert Oak, named after late night talk show host Stephen Colbert. And there was a video sent into his talk show to try to raise awareness nationally of Stadium Woods to hopefully get permanent protection. So they didn't end up cutting down the woods, but it still doesn't have permanent protection. So hopefully projects like mine and Alan's can help to gain awareness of how special it is and get it some protection. So I'm just studying the fungal diversity of Stadium Woods. I've collected specimens for the past year. I've been working on identifying them. I'm also posting them to iNaturalist. I have a project there. Then we hope to submit it for DNA sequencing, some of our specimens, since they're kind of hard to ID, and I'm not an expert in mushrooms at all. And we're hoping to find a comparison site. We're not going to be using the same comparison site as Alan. So we are hoping to find maybe a paper that's already been written on a secondary growth forest that we can use. If you guys have any paper suggestions, you could send them to me. So this is the protocol that we used for the mushroom collections. And if you want to donate any to our project, it's good to use this protocol because it's also the same protocol that the website needs for DNA sequencing. So just a photo of the entire mushroom, the cap and stem and everything, all the way down to the bottom and the top, with special attention to the spore structure. Any bruising or oozing, if you just gently press a stick or your nail across the spore structure, if it has a spore structure, you know, like a toadstool-like mushroom, if it changes color, it's good to note what color. Any odors that you smell, the date, the habitat that it grows on, and the coordinates. The coordinates can be found if you just take a picture with your smartphone and have your location on, they'll be in there. You can get it off with the app like Exif, or if you just post it to iNaturalist, it'll give you the coordinates of where you took the photo. It also has a feature where it suggests IDs based on just pictures, but you know, it's not always accurate, but if it looks accurate enough, you can include it. So more on the protocol, just if you want to take a cross-section of the mushroom, you should take it on a neutral background just so that we could see exactly where the bruising is. Like in this one, you can see that there's not a lot of bruising on the cap, but there's bruising in the stem and the gills. So it's good to note. Also, if it doesn't bruise, it might ooze. So just right here, you can see that I just had gently scraped across it with a pen, and some milky substance came out, and that's really good to note, especially if I can't get the specimen from you while it's fresh. For the sizing, you don't really need a scale for the photo like I have here. You could just have something that's universally sized, whatever's in your pocket, like a lighter, a pen, or I used a chopstick here. Also, if you collect any mushroom that's on wood or leaves, it's good to just collect some of the leaves or break the stick off rather than breaking the mushroom off, just in case you break any of the stem off. If it's out of dirt, it's good to scoop it rather than break it. So like you can see up here in the cross-section photo, there's a lot of dirt still there. You can brush the dirt off. I don't need the dirt, but it's good to try not to break any part of the mushroom off. So far we've found about 60 different specimens. Of the toadstool-like mushrooms, they're mostly boletes and they're hard to tell apart, at least for me. We found at least 2 amanitas, so that's fun. There's also a lot of lateral type mushrooms like in these photos here, or crust-like mushrooms. And we found about 20 different genera. So these are just some interesting photos that I have of the finds that we made. So there's some corals in here in the top right and in the lower left. There's some dead man fingers here in this corner. One of the bolete species and one of the amanita species. The turkey tail mushroom, go Hokies. And we found a lot of tiny little white mushrooms all over the forest, like these ones growing on just an acorn cap. And also this one was pretty common find. It's the hypomyces is actually a parasitic fungus that's attacking the bolete, and they specifically attack boletes. So over the summer, I was finding a lot of little like white blob mushrooms that I just couldn't even ID because they were attacked by this fungus. But, you know, hopefully we can find some interesting species for the herbarium and for the project. If you'd like to send any our way, you could just post them onto iNaturalist, and this is the QR code to our project. But I believe if you just post them in Stadium Woods, they automatically get added to my project because it just encompasses all of Stadium Woods, and if it's a fungus, it'll get added. And if you do make some collections, you could email me the rest of the protocols because iNaturalist doesn't ask for all of the protocols that we're asking for. You could also, if you find it and you don't feel like dealing with the mushroom, you can email me where you found it, the coordinates, just take a picture of it, and I could probably just walk there and collect it myself. So I just like to thank Beth Umberger for showing me around Stadium Woods, Alan Milby, the Massey Herbarium Lab Group members that have been really helpful to me throughout this process, everyone who's helped on iNaturalist, especially Beth, she's posted a lot, and the Native Plant Society for listening. Sorry.
Settings.
Any questions for Chrissy?
Chrissy, I don't know if I have a picture of it, but today we found one of the students on the privet, he was digging up, found little tiny little things, and they had like a little cup for, instead of having it was like a little cup that was up like that. They were little tiny. I posted it to iNaturalist, but iNaturalist didn't know what it was. So I don't know what it was, but it was on the privet.
Underground.
And they were underground. They were underground.
Oh, they were underground?
Yeah.
That's cool. I actually didn't find a lot of cup mushrooms myself, so I'll definitely look into that. Thanks. Let's see, there's a question. I'm not sure how long the project will continue. I'm still accepting collections, and I'll still walk through there sometimes trying to find a mushroom or two. But right now we're focusing on trying to find a comparison site because there's not a lot of papers out there of just fungal diversity in second growth forests around here. So if I have to go do the collections myself on a comparison site, it will probably last a good year or two more.
Thanks, Chrissy. This is Rebecca Paulson. I thought you might like to know that 2 of the people who got arrested in Stadium Woods in 2012 are watching this presentation, including myself.
Wow. That's really cool. You guys keep fighting the good fight.
We will. Thanks. And thanks for your work.
Thank you.
Any more questions for Chrissy?
I just might add that I took the course from Dr. Miller. I don't know if anybody knows who Dr. Miller was, but he was a superior teacher, and I was very fortunate to take that class in 1978. And we would go to Pipestem, and we would go to Pandappus Pond. And I'm telling you, I have never seen such, I haven't seen any morels in there, but there is a lot of diversity in that woods of mushrooms that if you were to go over to the where the stadium is, there's not like mushrooms like that. And if you go, that's what I keep pointing out to the students that are helping. And if you go over to that grassy area with the bamboo monoculture, there's a little bit of diversity over there, but I've not seen many mushrooms pop up over there. It is I don't know, is it like that in Center Woods? Does it have as many mushrooms coming up in there as they do at Stadium Woods? Do you know, Jordan?
I'm not sure. Alan, did you notice many growing out there when you were spending long hours out there?
I did. There was quite a bit of fungal diversity in Center Woods. It would be interesting for someone to go out there and look at that too. Yeah.
And Center Woods has it doesn't look, it's not at all, is Center Woods old growth, or has it been cut at some point, or?
It was cut at some point. There's some really old trees, just like Stadium Woods, but it was cut at one point, certainly. It's been a while since it's been harvested, but at one point it was cut.
Are there problems with invasives in there too?
To a less extent than Stadium Woods, I'd say.
I have a question for Christy. Have you contacted anybody from the Mushroom Club as to where you might find mushrooms or another site to do part of your research? Because those people, and there's dozens and dozens of people, will certainly know where all the good stuff is.
We have been in contact. I believe I'm a member of the Mushroom Club. I haven't got any paper suggestions yet, but I haven't been able to do a presentation for them yet. So hopefully after that, I can get more suggestions from them. I've just sent out like a mass email of my project to them.
Okay. Because there's some very good, very knowledgeable people there.
Seems like that's good for questions. So thank you again, Chrissy. Appreciate you taking the time tonight, and you did a great job. This was Chrissy's first research presentation, but I think she did quite well. So yeah, also wanted to thank Brenda for inviting us to come talk about our work tonight. So I'd invite you. What's that?
That was Mary Rhodes that initially.
Oh, right. Yeah, Mary and Brenda for making it possible. But I'd invite you all to the herbarium, but we're closed right now from COVID. So when things get better and we reopen, I'd encourage you all to come shoot me an email and set up a time to come stop by and see some of those old Orson Miller mushroom specimens and some of our bryophyte collections and other things. But yeah, thank you, Mary, and thank you, Brenda, and everybody for showing up. Appreciate your time.
We appreciate all three of you coming. So that was very interesting about all the different mushrooms and fungus and everything. So we'll have a brief chapter meeting now. There's two other presentations going on currently at the same time. One is the annual workshop of the Virginia Native Plant Society. It is part 2. Part 1 was on March the 2nd, I believe. And this year it's on climate change, which is the one that was supposed to be last year until COVID upset everything, of course. But at least they were able to have it this year. I know part 1 is already uploaded to the Native Plant Society website. I imagine part 2 from tonight will be uploaded there too. So everybody that couldn't attend will be able to view it in that. The other thing that's going on tonight is the second talk from the landscaping with natives that's being put on by the Planting with Natives in Virginia. And that's the one where they only had an initial 250 places and it sold out like within a day or two. well, it was only $10 for six webinars. And they finally bumped it up to 5,000. So if you're interested, you can still pay a measly $10 to watch all 6. And they're gonna, I think that includes another 6 in the fall for webinars and that too. And it's all aspects of landscaping with native plants, which is our main bent in that. Let's see. Next month, the April meeting will be hosted by me and Barb Walker. It will be on how to choose labels or plant signs for your plants in your gardens. I'll be giving different considerations to look into, and Barb will be giving some examples from Pandapus that she's used, and anybody else that wants to give me their experiences in that too would be good, about which sign that you like, why you like them, why you don't like them, how long they last, how legible they stay and everything. So things like that would be good. I understand that the Pandapus Pond Pollinator Garden is going to be restarting sometime this month. So if anybody's interested in helping out with weeding or planting or anything else there, contact Barbara Walker. And we're finally getting a second stretch of warm weather. It's going to be, I think it was 65 degrees today or something. So the skunk cabbages have been out for the last couple of weeks, and Carol Schwobel has given us a couple of locations where they already are. One was on the Huckleberry Trail on a new section between Price's Fork and Heritage Park along that boardwalk, the new boardwalk. And the other one is in the Deerfield Trail, where the bridge crosses the stream and going off to the left. But skunk cabbages are always interesting and everything. Let's see. Anybody have anything that they want to announce by any chance?
I am going to start looking for plants with local provenance to put in the woods that are not there. One of those are things like spring beauty, hepaticas, and some things like that. If you have a property that has local provenance, I would love to come collect some. What I would do is probably plant them in a bed in my yard first and get them bulked up before I put them there. I might put them straight in the woods. Dutchman britches, but there's no spring beauties in the woods. That's what surprises me, and they can colonize really nicely. So.
Well, there's both of those at Wildwood Park, but of course it's a park, so.
I got permission to collect from the woods in Christiansburg on town property, and I have some at the museum. I might transplant some of those over to the woods there.
Well, why don't you send me a list and I'll put it on the listserv then, and then we can remind other people that weren't at this meeting.
And I highly recommend that you go to Stadium Woods. When we wowed Jeff Kerwin by the amount of invasives that were gone, it should be really nice this spring with more Mayapples and all. So it has changed a lot. The woods is really opening up, and we are shocked at how many natives there are. Just lots of blackhaw and elderberry and things that are in the putty root we found. And also I found a cutleaf grape. If I've got a fern that's called cutleaf grape fern, I think is what it's called. Anyway, that was in 3 places. I was shocked. And there's some little apple tree in there that I'm hoping John Seiler will get cleared ID. But it's amazing, and it's opening up, and now it's starting to look like it should. So hope you'll come see it.
Anybody have any other announcements then? The board is going to be having a meeting later this month. We haven't decided on a date yet, but if you have any questions that you want discussed with the board members in that, just contact me and we'll add it onto the agenda. Anything from anybody else? Okay, if not, then thanks everybody for attending, and we'll see you next month if you're interested in coming then. Thanks very much to everybody that came. Okay, good night. And this will be recorded and put onto our YouTube channel.
Thank you, Brenda.
Thank you.
Okay.
Bye.
Thanks,
Bye.
everyone. Good night.
Night.
Thanks to our speakers.